Howdy! Welcome to Characterizing and Classifying Viruses, Vibroids, and Prions, Part 2. So this is the overview of viruses in Chapter 13, and it's the continuation of the first overview of viruses. So this is a cartoon. We went over this in the last video, overview Part 1. This is a cartoon of the coronavirus 2. So here's the name. Severe acute respiratory syndrome is the SARS part, and CoV is the coronavirus. And 2 is because they initially discovered a SARS-CoV. Maybe they didn't know there was going to be a 2, so they didn't call it a 1, but there was an original SARS-CoV. And then this is the strain. SARS-CoV-2 is the strain of coronavirus that causes COVID-19, which is the disease. And we went over this, but so this is the virus. These are these blue tulip-like things are attachment proteins. If these attachment proteins fit receptors on your cells, so the host cell down here, this is a phospholipid bilayer of the host cell. If the attachment proteins or the spike, aka the spike proteins on the virus, fit into these receptors on your cells, and you have another protein in your cell membrane that can activate the virus, that virus can get its RNA strand inside the cell, and then through a bunch of cool biochemistry, the virus can start making its own viral proteins, and then your host cell will lyse, and the viruses will get out. Sizes of viruses. So in the late 1800s, they thought that the causative agents of several diseases were actually smaller than a bacterium, and so they called them viruses, and that's Latin for poison. In 1892, they found that the filtrate from infected tobacco plants, here's an infected tobacco leaf, and if they got the filtrate from those diseased tobacco leaves, you could infect other leaves. And this is an old-timey electron microscope that they used to see the first viruses. So there you go, history. Okay, sizes of viruses. This is a red blood cell from a human, and it's about 10,000 nanometers in diameter. So from here to here, diameter, it's about 10,000 nanometers. An E. coli bacterium is about 1,000 by 3,000 nanometers, so 1,000 lengthwise, 3,000 nanometers, or sorry, 1,000 nanometers shortwise and 3,000 nanometers lengthwise. And then you come down here and you see that we look at the sizes of the viruses. So this is a cartoon of the smallpox virus. It's anywhere from 200 to 300 nanometers. So you can see it's way smaller than one of our cells, red blood cell, and it's smaller than a bacterial cell. Because you can see it in this cartoon up here, see, here's the smallpox virus in relation to this bacillus, and specifically it's E. coli. Tobacco mosaic virus, that's the one that caused those tobacco leaves, that disease. 15 nanometers by 300 nanometers. This is a bacteriophage. Remember a phage, or aka bacteriophage? They're viruses that infect bacteria. And then check out this little guy, bacteriophage MS2. MS2, oh, you know that's dangerous. Look how tiny it is, 24 nanometers. And then the polio virus that causes polio is about 30 nanometers. So our eukaryotic cells are really big compared to bacterial cells and viruses. How about some shapes of viruses? First shape we'll look at is helical. So remember, there's like the double helix, or a helical staircase goes around and around and around. And an example of that would be the tobacco mosaic virus. That's a helical virus. It says the capsomeres, remember those are proteins, little protein subunits. The capsomeres in a tightly wound spiral that form a tube around the nucleic acid. So these outside on this tobacco mosaic virus, those are the capsomeres. And then inside, it doesn't show it in the cartoon, but if you looked inside, you would find the nucleic acid of the virus. The next shape is called a polyhedral. So remember that from, I don't know when you had that, maybe junior high or high school, you went to geometry and talked about polyhedral. So polyhedral just means many sides. I think it's sides, but poly is many. So a common type of polyhedral is called an icosahedron that has 20 sides to it. And here's a cartoon, poliovirus. If you look at that, you can see these different faces. So there's 20 different faces or sides to the polio virus. And the next shape of a virus is called complex. So if you are a virus and you have a complex shape, that means you could have, for example, an icosahedral head, but then your tail would be helical. So you'd have those two different types of shapes. The smallpox virus, which is this guy up here, has several covering layers, but it doesn't have an identifiable capsid. So it doesn't have that outer protein capsid. There's another type of virus, picornavirus, and it's named for its physical properties, not necessarily its shape. Human immunodeficiency virus, so that's HIV, that causes AIDS, and the pox viruses, they're named for their clinical properties, not their physical properties or their shapes. So for example, human immunodeficiency virus is named for its clinical property, which is it causes immunodeficiency. And then in the clinic, someone who has got the smallpox virus has the clinical phenotype of pox marks on their skin. So if you are a virus, you could be named according to your shape, your physical properties, or your clinical properties. Okay. And these are some TEMs, that's a transmission electron micrograph of the helical tobacco mosaic virus, this guy. B is the polyhedral virus of the common cold. So here's a virus, here's a virus. C is a smallpox virion. So remember, virions are just viruses that are outside the cell. And then these are kind of interesting. The rabies virus, look, they kind of look like bullets. So they have this bullet shape. So this is a cartoon. Actually, this on the left, this is a scanning electron micrograph that is colorized of bacteriophage T4. And it shows its head, its tail fibers, and its base plate. And then on the right side here is a cartoon of the head, the tail region. So all of this stuff is in the tail region. These guys are tail fibers, and then this little ring type thing down here is called a base plate. And the head is a protein code of the virus, and remember it's made of those proteins called capsomeres. So we've talked about this with viruses, but viruses don't have their own cell membrane, aka their own phospholipid bilayer. But some viruses that are called enveloped viruses, they have their envelope is similar in composition to a lipid bilayer. And in this cartoon it shows here this part here, this is all lipid envelope. That means it's a phospholipid bilayer. And we've said this before, viruses, if they don't have an envelope, they're naked. And the envelope is acquired, the envelope of the virus is acquired from the host cell when the virus is replicating, or as the virus leaves the host cell, it will take some of that phospholipid bilayer with it and make a nice envelope for itself. So an envelope virus then has three components. An enveloped virus has genetic information. Now remember, that comes in the form of a double-stranded or single-stranded RNA or DNA. So that would have that nucleic acid in it. It has an enveloped virus has a protein capsid. So that's the protein surrounding the virus. And then if it's enveloped, it has that lipid layer or lipid bilayer that surrounds the protein capsid. Yeah, and so in this cartoon, they're calling it the nucleocapsid, but it's the same thing as a capsid. They just called it a nucleocapsid here because it's containing the nucleic acid. But it's, if you call it a capsid or a nucleocapsid, it's the same thing, and it's made of proteins. Okay, so in this cartoon, it shows an enveloped virus with a helical capsid. So here's phospholipid bilayer, that's the envelope. Here's the capsid. It's got a helical shape, so it goes like this. And then this letter B is an enveloped virus, so it's got the phospholipid bilayer. But it's got a polyhedral capsid. So here's one side of the polyhedron. This would be kind of like another side of the polyhedron. And then it's got the DNA or the RNA, the nucleic acid, inside the capsid. Okay, so this cartoon shows a virus, and it shows that it's got a lipid envelope. So here, let's take a look. It's an envelope, so this piece right here would be a phospholipid bilayer all the way around. So this is all lipid envelope. And then deep to the lipid envelope is in this cartoon they're calling it a protein envelope, but it can also be called the capsid. So these purple balls here is the capsid or the protein envelope. And then here's the nucleic acid on the inside, and it's RNA in this case. And then sticking out of the lipid envelope are these spike proteins. And now, instead of just generally calling them spike proteins, we're going to start being more specific. So in this cartoon, the spike protein, this green thing, is called neuraminidase, and this yellow cone-looking spike is called hemagglutinin or sialidase. So the influenza spike protein, so the virus that causes influenza, the influenza spike protein, hemagglutinin, is required, so this guy is required for attachment and entry into a host cell, and then this other spike protein, neuraminidase, is required for the virus to exit the host cell. So have you heard of the viruses H1N1? Well, that's where the H comes from, hemagglutinin, and the N is neuraminidase. This is just a list from your book that lists the characteristics of viruses and the characteristics of cells. So, for example, viruses do not divide, they do not grow. Cells do divide and they do grow. Viruses are acellular, cells are cellular, etc. Okay, so when you have a virus, you can classify it by the type of nucleic acid. So remember, that could be single-stranded or double-stranded RNA or DNA. The presence of an envelope or not. If you are a virus and you don't have an envelope, you are naked. The shape of the virus. So for example, the polio virus is a polyhedron shape. And then you can also classify viruses by their size. So, for example, bacteriophage MS2 is only 24 nanometers. And you can characterize viruses by their clinical manifestation as well, but that's not on this list. Okay, what about viruses in cancer? You probably know that cancer is just an uncontrolled proliferation of cells. So under normal conditions, non-cancerous conditions, divisions of your cells, say for example the cells in your GI tract that you have to grow and divide those all the time, that division is under strict genetic control. In our bodies, some genes for cell division are turned off permanently. That's like some of our neurons. Or you can have genes that inhibit cell division are turned on, so cells don't divide. You can have a combination of the two. Uncontrolled cell division produces a tumor, and if you are a malignant tumor, that means you can invade neighboring tissue and travel throughout the body. If you travel throughout the body, that's metastasis. You've probably heard these terms. The creation of cancer is oncogenesis, so genesis is creation. And it's interesting that viruses cause 20 to 25% of all cancers in the animal world, so us as human animals and other animals. And of human cancers, though, viruses cause about 15% of our cancers. And here's some cancers that are caused by viruses: Burkitt's lymphoma, Hodgkin's disease, Kaposi's sarcoma, and cervical cancer, all caused by viruses. Okay, so let's look at this cartoon. We'll look at the role of viruses in cancer. So here's a piece of DNA in the normal state. So this is the normal state. So you have this green piece here that's called a proto-oncogene. And then you have over here a gene for a repressor. So what happens in the proto-oncogene can become an oncogene. So proto just means before. So before you're a cancer gene, you're a proto-oncogene. And in normal conditions, this piece of protein, made from a gene and messenger RNA, it's called a repressor protein. And the repressor, when it's made, represses the proto-oncogene. So the repressor protein sits on the proto-oncogene. The proto-oncogene then cannot be transcribed because there's a blocker there. And your proto-oncogene stays a proto-oncogene. It doesn't turn into an oncogene. This is called the two-hit hypothesis, by the way. So the first hit, a virus gets into your cells and inserts this thing called a promoter in front of the oncogene. The promoter is a region of DNA that will turn on a gene. So you've got this promoter. The oncogene is now ready to be turned on, but you still have your repressor gene working. So your repressor gene is turned on, you make the protein repressor. The protein repressor comes over here, sits on the oncogene cannot be transcribed. Now the second hit, that's what gets you. So you're set up, you got a promoter region here in yellow, ready to turn on the oncogene to make an oncoprotein that will cause cancer. And then another virus, or the same virus, inserts its piece of nucleic acid, to DNA in this case, right into the middle of the repressor DNA, or the gene for the repressor. Now, you can make this repressor protein, but it doesn't work because it's not just this purple rectangle like it used to be. So it's got this piece of viral DNA in there. Now the repressor protein is mutated and it doesn't work. So that means that repressor protein cannot sit on the oncogene. It can't do that. So if it can't sit on the oncogene and the promoter's there, guess what happens? You turn on the oncogene. You transcribe the oncogene into messenger RNA. Then messenger RNA makes an oncoprotein, and then that protein goes on to cause abnormal cell division, and the result is cancer. this is called the two-hit hypothesis. So this, let's look at this in purple here. It says UV light, radiation, chemicals, viruses are all implicated in cancer via turning on these proto-oncogenes using this two-hit hypothesis or this two-hit method. All right. So how do you grow viruses in the lab? Obviously, we grow our bacteria in the lab. How do we grow viruses? So you have to grow viruses so you can do research and develop vaccines and treatments for diseases. But as you know, viruses cannot grow outside of a host. It has to grow inside a host cell. So you can use mature organisms. You could culture viruses in plants. You could culture a virus in an animal, but that causes ethical problems if you're trying to do experiments on your virus, but you use another living animal to grow the virus in. That has some ethical concerns, and it's also expensive to grow anything in a whole animal. But here's a good solution. If you're okay with this, you can culture viruses in an embryonated chicken egg. So here's the cartoon. Here's the little chicken embryo, and what you can do is you can inject your virus into the embryo. You can also inject your virus into these other parts of the embryonated chicken egg. You could, for example, you could inject the virus into this yellow stuff, which is the yolk sac. It's inexpensive culturing viruses this way. An egg is one of the largest cells, and this is all, the environment inside is all sterile, so there's no contaminating microbes there. And the viruses have a nourishing yolk. Vaccines are often made this way, but if you're allergic to albumin or eggs, you have to know if your vaccine was prepared in an embryonated chicken egg. You can grow a virus inside a bacteria. So remember, a virus that infects bacteria is called a bacteriophage. So you can take bacteria and the phages, you can mix them together in warm agar, and then you can pour them on these agar plates. So this is an agar plate. The phages released then, as it cools, as the agar cools, the phages infect nearby bacteria, and they destroy the bacteria, and all these little empty spaces here is where there used to be a bacteria, but the viruses killed it, and those are plaques. And then you can do a bunch of cool math and figure out how many viruses you have resultant from how many plaques you have. You can also culture viruses in cell culture. So here's a photograph, here's a cell, and you can grow viruses inside these animal cells. So let's just briefly look at other parasitic particles that we said we would talk about. We said we would talk about viruses, viroids, and prions. So a viroid is a small circular piece of RNA. There's no capsid, so it's not like a virus. It doesn't have any protein capsid around. It's just a small circular piece of RNA. It's infectious. It's pathogenic in plants. So it can, oh my gosh, it can infect my favorite food, the avocado. Be gone, viroids. You can also have viroid-like, so they're not viroids, but they're viroid-like agents that infect fungi. There's no known animal diseases, so nothing known to us that a viroid can cause a disease. The two potatoes here and here were infected with a viroid, and then here's a delicious, healthy, I'm thinking it's an Idaho potato. It looks delicious. Prions are the third of our infectious parasitic particles, so viruses, viroids, and prions. So here's a cartoon, of a cellular prion. It's got this shape, and then if it, well, it's actually, I take that back. This is just a cellular prion protein. It's got this shape, and then if it takes this infectious shape, it looks different and becomes a prion. So a prion's a protein infectious agent. A prion is not a nucleic acid. So remember, in viruses and viroids, those are nucleic acids. They're infectious particles, they're nucleic acid. Prions are not nucleic acids, they are protein. They're infectious proteins. And you can transmit them by eating infected tissue, transplantation, contact between infected tissues and mucous membranes. They can get into your brain, your spinal cord, eyes, skeletal muscle. These transmittable spongiform encephalopathies, it's like a group of conditions such as Creutzfeldt-Jakob disease and mad cow disease. You've probably heard of mad cow disease. These are all caused by prions.